extremely well known in the field, has accumulated most of the distinctions which are available for anybody working in our field of research. And in his website, he defines his main interest as saying that my research pursues a psychophysical approach to screening for memory malfunction in genetically manipulated mice. But I don't believe that. I think that his real interest is to create insightful and particularly incisive theoretical views which invariably create controversy and stimulate everybody to think in a different way. And I hope that he's going to do this for us today. So, Randy.
Well, thank you, Alex, but I'm going to disappoint you because I'm not going to do that, because Bill asked me to talk about the work that I did many years ago, and it wasn't my work, it was just a work of synthesis, on the structure of behavior, the lower level structure of behavior, and the intelligence and adaptability that emerges simply from the lower level structure of behavior. And so that's what I'm in fact going to be talking about. And there certainly isn't new modeling in this. And I think that this represents a synthesis of principles that are widely subscribed to by people who study the organization of action. I'm going to be talking about four principles, stressing four principles. One, that the mechanism is hierarchically organized. The second principle is that I will specifically illustrate is that higher level units impose patterns of coordination on lower level units, and they do this by selective potentiation and depotentiation. That is, they don't tell the lower level units what to do. They just, a little bit like an orchestra conductor, they just indicate when you're free to play as loudly as you can and when over here you're supposed to shut up. That's the fourth principle, and the third principle that I skipped over there is that it's an unusual kind of hierarchy, in that it, particularly in a structural organization, in that it's a lattice hierarchy, as illustrated here, as illustrated there by the diagram on the right. This was a point that Sherrington first stressed, namely that the units higher in the system compete for control over units lower in the system. This is a way of organization that would drive the Army nuts. I mean, suppose your platoon didn't belong to you and you were the sergeant, and you had to sort of compete with other sergeants for who it was that had authority over your platoon. But that's the arrangement that we find in the structure of behavior. So to put bite or teeth into this conception, we have to define units of behavior. And I don't think that we can improve upon the definition that Sherrington offered many years ago. And the pause there was that I realized the time on this lecture started back when I booted it up. So according to that, I'm 20 minutes into my lecture already, and that was a little startling. Sherrington said that a unit of behavior was an assemblage of neuromuscular elements that was sufficient to explain a naturally occurring behavior or movement or secretion. And he pointed out that the essential elements were an initiator element of some kind, that is, the signal that led to the observable behavior had to have a point of origin or initiation, that the muscles themselves, the effectors of the behavior, are almost never located at the site at which the signal that initiates the behavior arises, and therefore there have to be conduction elements, and finally there have to be effectors, the secretory glands and the muscles. And we can now follow the usual logic in science in distinguishing between elementary and complex units of behavior. This is exactly the logic that leads to the same distinction within chemistry, by noting that a unit is elementary if it cannot be experimentally decomposed into constituents that are themselves units of behavior. I hope you recognize that that's exactly how the chemist defines an elementary substance, right? Water is not an elementary substance because if you pass a current through it, you get oxygen and hydrogen. But oxygen and hydrogen are elementary substances because no matter how you bang on them, you can't get any other set of substances out of them. By contrast, a unit of behavior is complex if it can be decomposed into its constituents that are themselves units of behavior. So just to see the force of this, you might ask, for example, is the scratch reflex that Sherrington investigated at great length in his classic work, where the animal, in the spinal animal, usually the spinal dog or cat, raises its hind leg to scratch a point of irritation on the skin. Is that an elementary unit of behavior? And you can show by drugs and by various other things that no, that can be decomposed into two separate units of behavior that at least under experimental circumstances you can elicit separately. So you can get the animal sometimes to simply raise the paw to the irritated spot without actually scratching. And under the other circumstances, you can get the scratching reaction without the raising of the paw to the irritated spot. So the unit of doing this is the coordination of two separate units of behavior, one of which is bringing the paw to the point to be scratched, and the other is executing the scratching reaction. So can we, like the chemists, can we identify distinct kinds of elementary units of behavior at the bottom of this hierarchy? And again, I think the answer is yes, we can. And they're distinguished by their functional structures, and that's what I'm diagramming here. And the first of these units is, of course, the reflex. And in this unit, the point, remember, we in each of these units we want to say, okay, a unit of behavior has to have a source of signal, it has to have conductors, and it has to have effectors. So what is the source of the signal in the elementary unit, in the reflex? And the answer is it's the sensory receptors. And the conductors are just what Sherrington thought they were. They're the axons and synapses by which the signal that originates at the sensory receptors is propagated from one neuronal, from one cell element to the next within the nervous system until it reaches the effectors, which are typically muscles, but in the reflexes that Pavlov studied, they were secretory glands, salivary glands, and they can be tear glands or whatever you like. So muscles and exocrine glands are the effectors. The second kind of elementary unit is the servomechanism, of which the optokinetic reaction is an example. Almost all visual animals, if you rotate the visual field, the large visual field, either the whole animal if you're doing it with a fly, or the eyes if you're doing it with a human being or a rabbit, will rotate, tracking the moving visual image. And this has been this has been analyzed at considerable depth in the neurobiology of motor behavior. And it's a classic arrow-driven servo, and the functional structure of the servo is indicated here. And the thing I want to call your attention to is that if you just look at the parts of this mechanism that lie within the nervous system, the ones outlined in solid, the receptor elements, the conductor elements represented by the CTF, and the effector elements represented by the muscles that move the eyes, they're exactly the same as for the reflex diagram. So if you say, well, gee, if we're just looking in the nervous system, why are these distinct? Why would we distinguish between these units? Why would we claim that they have a different functional structure? And the answer comes when you investigate their quantitative properties. And there's a lovely contrast that brings out this answer very clearly. It's the contrast between the vestibulo-ocular reflex and the optokinetic reflex, optokinetic servo mechanism. The vestibulo-ocular reflex is a rotation of the eyes, also a counter rotation, that is also an image movement nulling rotation that occurs in response to the signal that originates in the semicircular canals when an animal rotates its head or has its head rotated. And if you ask what the gain is in that reflex pathway, that is, what if you have a given rate of rotation of the head, rotation of the semicircular canals, what is the rate of rotation of the eyes, counter rotation of the eyes? The gain is approximately 1, actually -1, because it's a counter rotation. If you do exactly the same experiment under open loop conditions for the optokinetic reaction, that is, you break the normal feedback that occurs between the movement of the eyes and the rate at which the image is slipping across the eyes. I want to make sure all of you are clear about what the feedback is. So in the optokinetic reaction, the image begins to slip across the retina, and then the eye is moved in the direction in which the image is slipping, right? So the movement of the eye partially nullifies the movement of the image across the retina that triggered the movement of the eye in the first place. That's what's captured by that dash feedback line there. Now, you can analyze this mechanism under open loop conditions. Basically, you have the image move across one eye, but you anchor that eye so it can't move, and you let the other eye move. So now, but the other eye isn't looking at the outside world. So now there's no feedback. Now you've broken the dashed line there, the feedback. And you can look at the open loop gain of this neurobiological mechanism, and it's on the order of 100 to 1. Right? So it's the gain in that loop is 2 orders of magnitude greater than it is in the gain in the vestibulo-ocular reflex. And the reason it is because, of course, this mechanism does depend on and take for granted the existence of the feedback that we have so artificially broken in this particular experiment. And in that kind of error-driven servo, the higher the gain in the forward part of the system, the lower the error, right? And what the system is trying to do is minimize the error. So the point is that the neurobiology has the properties that it has, namely a very high gain, precisely because the feedback is an intrinsic part of the unit. That's what's covered in that slide. The third unit that we can recognize, and at the time, back in the 80s when I was interested, this was still somewhat controversial within psychology. I think it's not at all controversial anymore, is that another elementary unit of behavior is the oscillator. And what distinguishes is the oscillator. And you see from the diagram of the functional structure of the oscillator that what distinguishes it is that sensory receptors are not the source of the signal that leads to the behavior that we observe, but rather the source is endogenous. It lies in pacemaker cells in the nervous system. And the oldest historical example of that is, of course, the heart. Harvey already knew that if you took out a heart, it beat while lying there on a table. And although there were repeated attempts over the years to try to explain this rhythmic beating by positing some rhythmic stimulus acting on the heart, it eventually became clear in the 19th century that the rhythmic beating of the heart arose from specialized cells in the sinoatrial node on the heart that are endogenously oscillatory, that are pacemaker cells. It took an astonishingly long time before people were willing to concede that similar cells existed within the spinal cord and lower parts of the brain, and drove the innumerable rhythmic behaviors such as stepping or scratching or breathing and so on. If you look at most behaviors, they actually have a strong rhythmic component, and I think the evidence, although this remains perhaps somewhat controversial, the evidence is pretty strong that in every case the rhythmicity originates from pacemaker cells. It does not originate exogenously. Another characteristic of oscillators, pacemaker, that is units driven by pacemakers, is something that was first clearly recognized and analyzed by the great German behavioral physiologist Erich von Holst, which is the presence of a phase shifting effect in response to timing signals, which is the key to understanding how it is one oscillator or pacemaker is coordinated with another pacemaker, and we'll see the force of that a little later on. But I want to stress this because it's a principle quite different from the ordinary reflex physiology. A pacemaker, what I call a phase shifting signal there, is a signal that comes into the pacemaker cell. But the curious aspect of this is that the effect of the signal on the effect of the signal on the behavior of the cell, on the behavior of the pacemaker, depends upon the phase of the pacemaker cycle at which the signal arrives. So if you send the signal into the pacemaker during one phase of its cycle, remember this is an endogenously cycling cell. It's cycling because of molecular processes intrinsic to the membrane of the cell. If you send the timing signal, the so-called Zeitgeber, into that cell during one phase of its cycle, the cell shortens its cycle. If you send it into the same cell, the same signal, at a different phase of the cycle, the cell lengthens its cycle, and this behavior is captured here in the phase response function, in which you'll see that what is plotted on the x-axis of this graph is the phase of the pacemaker cycle at which a timing signal arrives at that pacemaker. So it's a little unusual, and you're not plotting it's a funny kind of input parameter, because the parameter is the phase of the receiver's cycle. And what is plotted on the y ordinate is the shortening or lengthening of the subsequent cycle of the cell. And you see that for some phases there's shortening, and for some phases there's lengthening. And to understand the functional significance of this, all you have to do, if you've ever had experience in a marching band, however amateurish, and mine was quite amateurish, but I was in a high school marching band, and you reflect on how it is you stay in phase with the drum, right? Well, if you hear the beat of the drum an embarrassing fraction of a second after your foot hits the ground, then what you do, as inconspicuously as you know how, is lengthen your, is hasten your next stepping cycle, right? Whereas, I'm sorry, I got it backwards. If you, I had it right the first time. If you hear the beat of the drum after your foot hits the ground, right, at that phase of your stepping cycle, then that means you're stepping too fast, or you just stepped too fast, and you lengthen the next step cycle. Right? If you hear the beat of the drum shortly before your foot hits the ground, then what you do is shorten the duration of the next cycle. And it's this process of phase-dependent responding to the beat, to the time signal, which is the drum, that it keeps you in step with the rest of the gang. And that's exactly what's being plotted here in the phase response function. And the important thing to realize is that seemingly rather complex process is implemented at the cellular level. That is, you see this phase response function is the response of an individual pacemaker cell to a timing signal arriving at different points in its cycle. So that brings me to the fourth of these elementary units of behavior. And this was not, so far everything I've said was in my book in 1980. It's a little embarrassing to be talking about material that goes back that far. But there has been progress since then, though I've not been keeping up with it as much as I used to. And one of them is the emergence, this is particularly due to the work of Steve Lisberg, of the realization that some servo mechanisms were not simple error-driven servos. There's no standard term, but they're often called latching servos or sample and hold servos. And they, again, there's a feedback indicated by the dashed line from the output action to the input velocity, but this isn't the basis of a continuous correction in response to the resulting error signal. Rather, the system makes an initial estimate of the velocity. You see this system in smooth tracking, and I'll show you one of Lisberger's records in a moment to describe what I'm talking about. Rather, the system makes an initial estimate of the rate of movement of the object that it's trying to track. It stores that estimate in a memory element, and it uses that estimate to drive the movement of the eyes. And it doesn't make, when it corrects its movement, it does so in a step-like fashion. That is, if its original estimate proved to be erroneous, so that after a while there's a growing discrepancy, that is, the object being tracked is moving out of the fovea, then there will be a step-like change in the velocity of the eye. And here's an example of the from Lisberger's work of he has many experimental records that look exactly like this, although if I recall correctly, I basically sat down and drew this. But what is being plotted here is the angular position of a small target that the subject, which was monkeys, are tracking. And the target is stationary until you see the sudden elbow in the line, at which point it begins to move at a constant velocity. And you see, not surprising, the dashed line is a plot of the angular position of the eye. And you see, not surprisingly, that there's a reaction latency. The eye does not react to this immediately. And when it does, it first begins tracking at the velocity of the target. It's a little hard, it's sort of obscured by the dashing. And then there's a saccade to foveate the target while the eye is already moving. So the eye is moving with the target, but the target is out of the fovea. Right? The eye makes a step correction to foveate the target, and now the eye is moving at exactly the same angular velocity as the target. Now that's impossible in an error-driven servo. That is, one of the characteristics of an error-driven servo is there's always a residual error, because it's the residual error that is driving the reaction. That is, it's this goes back to the high gain point that I was making earlier. In a simple servo, it's the error signal that is driving the reaction. And so you can never get the action matching the stimulus. But Lisberger has showed over and over again that when it comes to smooth pursuit, you can get the eye exactly, the angular velocity of the eye essentially exactly matching the angular velocity of the target. And by that and a variety of other manipulations, they've shown that that's because the eye is being driven by a remembered estimate of the rate at which the target was moving. So this is clearly a different kind of servo mechanism from the this simple error-driven servo. And the fifth elementary unit that I will mention is also relatively new, although evidence for it has really been around a long time. In fact, I'll show you two examples of evidence for it, and both of them go back quite a long ways. So this elementary unit of behavior requires a variable coordinate transformation. Now, almost all behavior actions require a coordinate transformation because the action is not conducted in the same framework as the stimulus that gives rise to the action, even in the case of the optokinetic and vestibulo-ocular. Well, consider the vestibulo-ocular reflex to take a particularly simple example. The signals that drive the reaction are the signals that come from the semicircular canals in the ear. And those semicircular canals are arranged in three planes, three orthogonal planes, as you might expect if you believe in the power of geometry, to pick up three orthogonal possibilities of rotation. That's the coordinate framework in which the planes of those three semicircular canals establish the coordinate framework in which the sensory signal originates. But the motor signals, the signals that go out to the muscles that drive the eye, are working in a coordinate framework established by the planes in which the pairs of muscles that move the eye operate. And those planes are not the same planes as the planes of the semicircular canals. Right? So in the course, one of the things the nervous system has to compute is a coordinate transformation in the process of going from the sensory input to the motor output. That, however, is not what leads to the distinction here in this fifth elementary unit of behavior, but rather it's the many demonstrations that there are a great many elementary units of behavior, and I'll show you two examples, the saccade in humans and monkeys, and the scratch reflex in the spinal frog, in which the coordinate transformation that has to occur between the sensory input and the motor output is context dependent because the two frames of reference are themselves constantly changing. And this will perhaps be most clearly illustrated by this double saccade experiment that was pioneered by David Sparks and Nelson back in the late 80s. So in this, people had imagined, up until the time of this experiment, that the saccadic reflex was a simple kind of reflex that I've already described. That it was known to be a reflex, right? So a saccade occurs when something is suddenly illuminated outside the fovea, and the eye steps in a single ballistic movement to foveate the target. And we know it's not a servomechanism because although, of course, under ordinary circumstances, it's true that as the eye moves, the image of the target moves across the retina. It had long been known that you can shut off the target source as soon as the eye begins to move so that there is no movement of the image across the retina, and this has no effect on the course of the saccade at all. Right? So that the trajectory and stopping point of the saccade have been planned before the saccade begins, and they go to execution independent of the feedback. So although, this gets back to a point that John and I were talking about at the end of his talk, although feedback is present, feedback is not in fact driving the unit when it comes to a saccade. But it had been imagined that this was a particularly simple reflex of the kind you can fairly readily imagine. So a spot of light falls 20 degrees, let us say, to the left of the fovea. In what follows, I'm going to ignore the fact that the lens left-right reverses, right? Because the reversal is too confusing. So I'm going to, in actual fact, a light out there at the left falls on the right side of the retina. I am deliberately ignoring that effect. And conceptually, we can imagine it falls on the left side of the retina. For the following point is elementary. So you have the you can imagine that while you have this signal that comes from 20 degrees away from the fovea, and that leads to the planning, to the programming of a saccade that jumps the eye 20 degrees to the left, right, which is a phenomenologically accurate description of what goes on. But the Sparks and Nelson experiment shows that what's going on in the nervous system is considerably more complex than that story would lead you to believe. And to do this, they took advantage of the fact that there's about 150 millisecond delay between the time when a flash appears on the retina and when you get the answering saccade. And that's fact number one that you have to know. And the second thing is that if you deliver a second flash at a different location during that 150 milliseconds, after the first flash has occurred, but before the movement in response to the first flash has occurred, what you then get is a double saccade. You get a saccade elicited to the first flash, followed immediately by a second saccade directed toward the second flash. And they realized that this opened up a very interesting possibility, which I'm trying to illustrate by this diagram here. And it shows that the system is doing a variable coordinate transformation, taking into account the change in the frame of reference between the time at which the stimulus was received and the time at which the action was executed. So in this experiment, the first flash falls on the, now here in the diagram I can't ignore the geometry of the situation, so the left-right reversal is present in the geometry here. So you see that with a target light to the right of the optic axis in the left of those two diagrams, the flash falls on the left of the retina, right? But now suppose that earlier on, we have delivered a first flash. That is, suppose that that light is the second of the two flashes. And suppose that earlier on we have delivered a first flash from a source that lay further to the right of that light, right? So that in making the saccade to the first flash, the optic axis of the eye is swept past the light that generated the second flash. Now it's very important to bear in mind that light is no longer on, right? There is no image on the retina from that second light, right? This movement that changed the frame of reference occurs after the second light was extinguished. But now the question is, what is the saccade that you will see to this light here? And notice that whereas at the time the stimulus was received, the light lay to the right of the optic axis. But at the time when the movement is to be executed, the light, no longer visible, lies to the left of the optic axis. And what you in fact see is an appropriate saccade. That is, the saccade is not rightward as it would have been if there had been no intervening movement, but rather leftward. In other words, the nervous system has calculated the change in the geometric relationship between the optic axis and the source due to the first movement, and that coordinate transformation appears in the second one. And this is in a saccade, right? So saccade is basically as simple a reaction as we know of. And you see a similar point in a famous experiment by Birkenblit and Fuchsen and Feldman. Russian investigators. And this is in the spinal frog. So the frog, we've eliminated the brain of this frog, right? What we're seeing here is the integrative capacity of the spinal system, of the spinal cord of this frog. And with the forelimb of the frog in this position, they introduce a drop of acid on the skin of the elbow up here. And if the forelimb is in that position that we see up here, then the hind leg moves up all the way here and executes a scratching reaction like that, right? But if you passively move the forelimb of the frog down into this position, changing the relationship between the sensory framework, which is this one, and the motor framework, which is defined by the angles of the joints of the hind limb, which is what the muscles are controlling, then you get, in the spinal frog, an entirely different movement, scratching movement of this leg. So again, the spinal nervous system has computed the variable coordinate transformation between the input and the output. So you see a lot of complexity very low down in the system. You see a lot of integrative action at the level of individual cells and a lot of integrative action in spinal circuitry. Second point, so then now I'm moving to the establishment of complex units of behavior and how they impose a pattern of coordination on the lower level units. And one of the key ideas here is the idea of a central pattern generator, which was, like most of these, was originally quite controversial. The ethologists, among others, contributed to this idea, but has now been shown over and over again at the single unit level. And we've gained considerable insight into the mechanisms underlying these central pattern generators. So a central pattern generator is something that produces a patterned motor output in the absence of any patterned sensory input. The Sherringtonian, and to a strong extent the behaviorist tradition, always assumed that the explanation for patterns in the behavioral output had to lie in patterns in the sensory input. Yeah. It's kind of the brain as passive telegraph system. And the idea of a central pattern generator was, no, you can have complex patterns in the output that have purely endogenous origin. And we now understand that at least many of those central, first of all, that's clearly true, and second of all, that it's not as mysterious as it appears once you appreciate the existence of these pacemaker cells, which are themselves an endogenous source of activity that can appear in the musculature, that can be transmitted to and thereby be manifest in the musculature, and that these oscillators can be coordinated one with another to create complex patterns. And that this is one of the keys to understanding the enormous variability that we see in behavior. One of the challenges of understanding behavior, one of the major challenges, is that it's extremely variable. The more minutely you analyze it, the more you realize that the animal never does quite exactly the same thing twice from one occasion to the next. So the animal's constantly accomplishing approximately the same thing, but with muscular actions that vary from one occasion to the next. And one of the mysteries is to understand how can the muscular reactions be so different and the end result be so constant. And one of the illuminating studies of this is the study of gait control in the cockroach, because the cockroach has, like all insects, has 6 legs. And so you see very complicated gaits, and they're extremely diverse. And here's the gait that you see in the slow-moving cockroach that the amateur observer is not so likely to see. And the cockroach begins with a step on the right side. The black plots the point at which a leg steps forward, right? So the right rear leg steps forward, then the right middle leg steps forward, then the right front leg steps forward. In other words, the cockroach has moved all three legs on one side, starting at the back, then the middle, and then the forward, without moving any leg on the other side. And then you see the pattern begin on the other side. The left leg steps, then the left the left hind leg steps, then the left middle leg, and then the left front leg. So here's the gait that the nighttime kitchen raider is much more likely to observe in the cockroach, and it could hardly look more different from the gait that we just saw. Now, this is the well-known tripodal gait, and that's because the animal is stepping in alternate tripods. So now we notice that the right rear leg and the right front leg and the left middle leg are all stepping together in this gait. And so that's during that period when that tripod is stepping, the other tripod is supporting the roach. And then next thing that happens is the next tripod steps. Now the right middle leg and the left front leg and the left rear leg step. And so that's, and there are all variations in between. There are literally hundreds of gaits, stepping sequences that you can see in the cockroach. Well, the first big step toward realizing what was going on, as usual, was careful behavioral analysis due to Wilson and others. And Wilson realized that there's much more constancy between those gaits than strikes the eye at first analysis. He noticed that in both cases and in all the other gaits, there was in fact a wave of stepping that started at the back and went forward on each side. And you can see that here in this rapid one, and here it's obvious in the slow one. Right? He realized that in every case, the legs on opposite sides of this body were always 180 degrees out of phase, whether they were running in the slow. So you see that here. Here's the right rear leg, and here's the left rear leg, and here's the next right rear leg, and it's 180 degrees out of phase. That is, the step of the right rear leg falls halfway between the two sequential steps of the left rear leg. And the same thing is true down here. Okay, that's another motor constancy. Another thing that Wilson noticed was that this lag between the stepping of the right rear leg and the stepping of the right middle leg remained constant. Similarly, this lag here between the stepping of the middle and the stepping of the forward leg. What changed was the cycle period, how quickly the cycle began all over again. So this led, I'm assembling here the elements necessary to understand this, and let me, because I'm running out of time here, let me quickly go on to give the contemporary view of what we think the structure of the underlying mechanism looks like. So we know from direct electrophysiological evidence that there are pacemaker cells for each leg. So there are 6 sets of pacemaker cells, and they establish the basic stepping rhythm for each leg. And we know something about how they activate the opposing populations of motor neurons that trigger the actual step itself. The key to the thing, though, is this higher level. This is now an example of a complex unit of behavior. We've got the elementary units of behavior, namely the oscillators, and they're being coordinated by a higher level circuit. And the keys to the higher level circuit are, first of all, the couplings, which are indicated here. So here we have the fixed lag coupling that was evident in the behavior. That is, this neuron at the back of the animal, this pacemaker, at the point in its cycle is sending a signal to the pacemaker for the leg in front of it that entrains the oscillation in this pacemaker so that it runs a fixed amount of time behind this one. In other words, it peaks at a fixed amount of time. If the time is fixed, the phase cannot be fixed, right? So you can either fix the phase or you can fix the time, and this coupling fixes the time, which is why this is called a fixed lag coupling, and we have a similar coupling here. Whereas these oscillators that lie, these pacemakers that lie on opposite sides of the ganglion near the midline, are coupled with fixed phase couplings indicated here. And finally, we have we have large command neurons that descend from a higher level ganglion in the rostrum and that establish the common period of all these pacemakers. So you have a single signal that speeds up the rate at which the animal's stepping. And this system generates all the different gaits that are observed. And I don't have time for that. So I think I've already stressed the importance here of this as an example of how a simple mechanism with a fixed structure, not a plastic structure, at least in the way in which learning people understand plastic, can generate a baffling variety of gaits, all of which are functional, all of which are adaptive. So the final thing is how potentiation and depotentiation. What's my time here?
You have 4 minutes to the end of the end.
Okay. So damn. I'm damn because I very much wanted to leave questions. But anyway, so here's an example of a lovely, it's been known for a long time. Well, part of it's been known for a long time. Again, this is in the spinal cat. Right? So again, we're just dealing with the integrative capacity of the nervous system in which the brain is gone completely. If you bring the hind leg of the cat up against the tabletop like this so that you touch the dorsum of the paw, the leg lifts up and places on the table. This reaction has been known since the 19th century. So this is a flexion reaction of the hind leg in response to a touch on the dorsum of the paw. If you now set this spinal cat up walking on a treadmill, you have to support the cat because the cat no longer has a balance reaction. But if you support the cat will walk on the treadmill. And now you reach in and you deliver exactly the same stimulus to the dorsum of the paw, but you deliver it during a different phase of the stepping cycle while the cat is standing on that leg. And now you get exactly the opposite reflex reaction. That is, you get an extension reaction rather than a flexion reaction. And you can understand when you watch this and reach in with a stick touching the dorsum, you see why the change in this. If you're standing on a leg and you're struck by something, a rolling stone in the path or something like that, the worst thing you can do is retract the leg, right? And this is like adolescent boys go up and hit the backs of the knees of other boys because it makes you lose your stance. So the worst thing you can do is flex. What you want to do is extend so that you get the weight, the support off of that leg as quickly as possible and onto the other leg. And that's in fact what we see here. Conversely, if you're during the swing phase of the cycle and you hit something, right, now what you want to do is not extend. That's going to make the problem worse, but flex to move the paw over the obstacle. So these reflexes are stumble-preventing reflexes. But the point is you have two opposing stumble-preventing reflexes, and each one is functional under a different circumstance, and the pacemaker level of the system controls by potentiation and depotentiation which of those reflex pathways is available to become active. That's why we use the term potentiation at any given moment in the pacemaker cycle. And in doing so, it ensures that the stumble-preventing reflexes, which are one elementary unit of behavior, are coordinated with the pacemaker, with the stepping cycle, which is another elementary unit. And on that note, I end. And I apologize for not...
Well, Randy, this seems really neat. You know, especially the part about the cockroach. But you know, if cockroaches will run away from a threat, but you know they'll approach something like a dark place or food, and I'm having trouble making the leap from talking about these elementary units and so on to actual behavior of that sort. I wonder if you could help me out a little.
Well, I only had 40 minutes, right? In my book on the organization of action, of course, that's just what I attempted to do. That is, I showed, I never, in the talk today, I never made it to the hypothalamus, right? Until you get, I never got past the top of the spinal cord. Until you get to the hypothalamus, you don't get to integrated behavior. But the principles work all the way up. Right? That was the main, there's a short account of this in the American Scientist called From Muscles to Motivation. And it emphasizes the fact that motivation acts by selective potentiation and depotentiation. That motivational states turn on, make certain reactions, they potentiate certain reactions and depotentiate others. One very elementary example of that is the lordosis reflex in the female rat. Right? When at a certain point in her estrous cycle, if in a rat lab where they want to know the cycle of the females, they go through the lab, opening the thing, they reach in and squeeze the flanks. If she tries to bite you, she's not halfway through her estrous cycle. If she presents her genitalia, then she's in her estrous cycle. Right? So, in response to this sexual stimulus, because when the male mounts the female, he usually clasps her flanks, you get two opposing reactions depending upon the motivational state of the female. One being the presentation of the genitalia, and the other being turning around to bite this bastard. And you can you can construct most of sexually motivated behavior, at least in the rat, just by following that principle.
I think we have time for one more question. If anybody's in a hurry to leave before the next speaker, you can start going now.
Yeah.
Any other questions? I have
My impression with people is that if you deform the tendon to the side bone, from the shin to the thigh, you actually extend. I mean, this is a stumble dimension. I'm just curious.
Yes, no, that's true, but that reflex too is completely absent during certain phases of the step cycle.
Could you go on and which part? I mean, when you're okay. If it's swinging forward
during the, if I think it's during the stance phase, but I would have to.
Okay.
But I once went looking for reflexes that weren't subject to potentiation or depotentiation. That is, whose operation was fully automatic, that they happened independent of higher level activity. And basically I couldn't find any in the literature, any reflex. And I focused particularly on the stretch reflex because it's so often, I can steer you to the references that show that the stretch reflex pathway is completely inoperative at certain phases of the stepping cycle. And that